What's up, guys? This is Jahangir from MedPorium, and today we're going to be talking about protein isolation, purification, and also separation. So this is going to be specifically for the MCAT biochem section, and it's also good to be an expert in these topics that we're going to be discussing, since the MCAT usually pretty much tests on these subjects that have been shown up in most of the exams. So first off, let's start with protein isolation. So I'm just going to be writing isolation over here, because if we want to be able to separate our proteins or even purify them, we have to be able to isolate them. So protein isolation, this can come from body tissues or cell cultures, and this can occur through lysis or homogenization. Now this includes crushing, grinding, or blending of the tissues to homogenize them. I'm sure you guys are all familiar with the centrifuge and what it usually does. So for example, if we want to get a CBC, a complete blood count in the clinic or in the hospital, we're going to be taking blood from the patient, and with our centrifuge, we are going to be separating the plasma from the erythrocytes and all of those proteins that are part of this plasma over here. So through our centrifuge, we're actually able to isolate proteins from those smaller molecules. So we're able to isolate the proteins in the blood from the erythrocytes and that connective, which is also known as a connective tissue. So that's one method and one way we could do protein isolation. Now let's talk about electrophoresis. Now, electrophoresis is a way we can separate and purify our proteins, and this is when we are going to be eliciting our proteins or any sort of substance that we're using through an electric field. So that electric field is going to be placed on the whatever we're using. Let's just say since we're talking about proteins, let's just talk about proteins for this. So since we're subjugating these proteins in an electric field, our proteins are going to be moving based on their net charge and size. Now over here we have two different electrodes. We have the negative electrode and we have the positive electrode. Now when we're talking about electrophoresis, our negative electrode is going to be our cathode. So cathode, and our positive electrode will be known as our anode. Now, this might be different if you remember from general chemistry talking about the galvanic or voltaic cells, where we had the cathode being positive, anode being negative. In electrophoresis, that is going to be the opposite. So cathode is going to be the negative side, anode is going to be positive. Now, let's say we had some charged proteins. Let's say I'll just put like a positive over here, and let's say we have a negative guy over here. So we have positive protein, negative protein as their charge. So the positive protein will be moving closer to the cathode, so it'll be staying a little bit over here, staying on this side right here. There's not going to be much movement since it's attracted to the cathode, while our negative protein is going to be moving towards the anode, since opposites attract. We have a positive moving to the negative, or staying at this negative side, while our negative is going to be moving towards the positive anode. Now, there is one equation that you don't really have to memorize this for the MCAT, but it's still good to know, where we can measure the migration velocity. So migration velocity is going to be determined by our electric field strength, denoted as E. Then we have our net charge, which is going to be Z, and this is going to be over our frictional coefficient. So this is just everything you need to know for electrophoresis. And now what about that gel that we're using, since this is known as gel electrophoresis? Well, the gel that we're going to be using is polyacrylamide gel. So polyacrylamide gel is going to be used for our protein electrophoresis. Now, it's slightly porous and it solidifies at room temperature, but the main thing you need to know is that small, high-charged molecules will move faster in our gel electrophoresis in this gel, and large neutral molecules will move slower. So there's different ways we can there's different types of electrophoresis that we'll be talking about. The first one, and let's create a subcategory over here. The first one is going to be known as native, and then PAGE. PAGE stands for our polyacrylamide gel electrophoresis. So that's what PAGE stands for, and native is this specific method of our gel electrophoresis. And this is in which our protein will be in its native form. So I'll just do a bulletin point and I'll just put native. So our protein is going to be in its native form. There's no denaturation that's going to be occurring. So our protein is going to be separated by its native form. And our protein is going to be separated by a couple of things. We're going to separate by size, charge, and we're going to be separated by shape as well. So because there is no denaturation that is going on, we can separate our protein in its native form with these characteristics. And one thing you'll know with the MCAT and its lingo is that when you stain proteins, when you're going through electrophoresis, that's when you denature them. So any sort of staining to our protein with any sort of chemical will denature our protein. Now, what is a method of gel electrophoresis in which our protein is denatured, and how do we sort of call that? Well, one of them is known as SDS PAGE. And this is the main one you have to know. It's just these two for now. SDS stands for sodium dodecyl sulfite. I always screw up with those pronunciations, but just remember it's just SDS, and it's a chemical that when it is introduced, it's a detergent that disrupts non-covalent interactions, and all you really have to know is that this denatures our protein. So I'm just going to put bulletin point, and this is denatures our protein. And this is when we're going to be separating our protein just based on size. So in SDS PAGE, we're going to be separating our protein just on size. Now there's two different types of SDS PAGE. There's non-reducing and reducing. Reducing SDS PAGE is going to be separating those, or is going to be breaking those disulfide bonds. So if there's any disulfide bonds created by those cysteine amino acids, if there's any disulfide bonds, then our protein will be separated according to those cleavages in those disulfide bonds. But just the basics that you have to know is that SDS PAGE is when we're going to be separating those proteins when they are denatured, and we're going to be separating them based on size. The last method of separation we need to understand is isoelectric focusing. This is when we're going to be separating our proteins based on the isoelectric point. Now, the isoelectric point is a pH at which our amino acid will have a net zero charge. Now, for acidic amino acids, our isoelectric point is going to be low, so I'll put acidic. And for basic amino acids, our isoelectric point will be a little bit higher for basic. And as you should know by now, our acidic amino acids, when they're put in a physiological state, they will retain a negative charge. So these will usually have negative charge. And for our basic amino acids, for the most part, when they're in a physiological pH, they're going to have a positive charge. And as you'll notice with isoelectric focusing, as pH increases, our amino acids that travel further as pH increases, so let's just say that this is pH and this is the trend that's occurring. As our pH increases, the proteins that have a higher isoelectric point will be moving farther, while our proteins with a lower isoelectric point will stay relatively over here, moving towards that lower pH. And our proteins will continue to move until they hit that isoelectric point, until they hit the point in which they retain that net zero charge. So now let's talk about chromatography. Now, chromatography is another high yield topic that we have to know in the biochem section, and this is where we can isolate our proteins based on, actually a lot of characteristics. So one of the most common ones that you'll see in the MCAT is going to be TLC, thin layer chromatography, or also known as paper chromatography. The setup is usually like this, where with this kind of chromatography and also column chromatography over here, we are going to have a mobile phase and we're going to have a stationary phase. So the mobile phase will usually be some sort of solvent, and for the most part, by default, the MCAT will tell you that the stationary phase is going to be some sort of silica gel. Silica usually is going to be polar, and since the stationary phase is polar, our mobile phase is going to be non-polar for the most part by default, unless it clearly states something different. We are going to assume that our mobile phase is going to be non-polar and our stationary phase is going to be polar. Now, when we subjugate these proteins in a thin layer chromatography, The proteins that travel farther will be less polar compared to the proteins that stay with the stationary phase, because they're more attracted to that stationary polar phase compared to the proteins that are more attracted towards that mobile phase. The mobile phase is pretty much traveling them, pushing them out. So if they're more attracted to that mobile phase, they'll move a little bit more, while if they're attracted to stationary phase, they'll pretty much stay where they are with little to no movement. And that's when a protein like, let's say this guy over here, the blue guy over here, is going to be more polar than the red or the green dots over here. And another way you can find out how far your protein has traveled is through the retention factor, or RF, which, it's really simple. All you have to do is just find the total distance, and it's going to be the distance traveled by the middle of this dot over the total distance. And that's just easy math that we don't have to get into. But TLC is going to be separating these by polarity, and column chromatography also does the same thing. Column chromatography is this guy over here. Setup is a little bit more different, where we're going to be using gravity to elute these proteins, and we're going to have our mobile phase right here. Mobile and stationary phase will be right here. Stationary. Pretty much same thing over here, but the proteins will be eluted. The only difference is we're using gravity. And let's just say we had something like this guy over here. So we have this protein right here and this one right here. So the proteins or substances, or whatever you want to call them, that elute last, they are going to be more polar, because our stationary phase, by default, we're assuming that it is going to be polar. So anything that is eluted last is going to be more polar. It's going to be the most polar because they stayed with the stationary phase, while the mobile phase, we're assuming that is going to be non-polar. So anything that is attracted to the mobile phase will elute out as fast as possible. So that's TLC, that's column chromatography, and both of which are separating based on polarity. Now, what if we talk about ion exchange, size exclusion, and affinity? Well, with ion exchange, which I'll change my color over here, I'll do ion. Let me actually increase the font right there. We're going to talk about ion exchange, and there's two types exchange. So the first one is going to be cation exchange. So cation. And with our cation exchange, we're basically assuming that our protein is going to be positively charged, and our column will be negatively charged. Now with the next one, which is going to be anion, we are going to have our protein being negatively charged and our column being positively charged. So this is, with ion exchange, we're basically separating our proteins based on charge. So I'll put charge over here. Next one we have is size exclusion. So let's change my color to this. We have size exclusion. So by the title, you can guess that we're separating these proteins based on size. Now, with our size exclusion, one thing to note is that our smaller proteins, these smaller proteins will actually stay. They won't be eluted as fast. So I'll just put, just stay, just to put a word out there. While our larger proteins will be eluted first. So that's the number one thing you need to understand with size exclusion chromatography, is that the large proteins will be eluted first. And that's the number one thing you have to know for that. And finally, we have affinity chromatography. And affinity chromatography is going to be separating our proteins, let me change my font over here, based on binding affinity. So it's going to include questions like if we had a his tag or anything like that, where our
proteins are going to be binding to a specific molecule, and only the proteins that can bind to this specific molecule will be retained. So that's the thing with affinity chromatography. It's actually one of the ways in which we can purify our protein, and this is where binding affinity will come into play with those his tags or resins, where that specific molecule will be able to bind to that protein. But that's pretty much it for chromatography.